Welcome, everybody. My name is Sean Hennessy, and I'm delighted to host this webinar on drug-drug interactions. I'm going to be co-presenting with Charlie Leonard, but we'll be doing it in a tag team fashion. So today I'm going to be going over the study methods to study the health effects of medications, which is basically an overview of pharmacoepidemiology methods. And then on June 22nd, Charlie Leonard's going to present pharmacoepidemiology methods that are specifically applicable to studies of the health effects of drug-drug interactions. And then on June 29th, Charlie is going to present an example of a recently completed pharmacoepi study of drug-drug interactions, and that's looking at the combined effects of sulfonylurea glucose-lowering drugs together with lipid-lowering drugs. In terms of ground rules for today, so this is our first one of these webinars, so I'm new at this. I hope you'll bear with me. There are a lot of people on the line, and more people will be dialing in. So if people have comments or questions, I'd like for you to type them into the chat box. You can either direct them to me or direct them to everybody, whichever you like, and then I'll get to them either as the presentation goes on or at the end. I wanted to share with you some of my motivation and Charlie's motivation for putting together this webinar series. So Charlie and I and others here at the University of Pennsylvania have been conducting pharmacoepidemiology research on drug-drug interactions for the past 10 years or so. We've published lots of papers in the area, but we found that our papers don't seem to be having the impact that we would like them to have in terms of affecting prescribing and affecting the drug-drug interaction compendia and knowledge bases that are used to trigger alerts in clinical decision support systems. So Charlie and I put our heads together to think about how to address that, and we talked to some folks that curate some of the compendia and knowledge bases, and got the idea from that of trying to provide some educational webinars on pharmacoepidemiology methods, which are methods that many of us aren't familiar with. I know that a lot of the people that curate compendia and knowledge bases have a lot of experience with pharmacokinetic studies and the principles of drug-drug interactions. And our intent here is to share some of what we know about pharmacoepidemiology and the methods of using pharmacoepidemiology to study drug-drug interactions. So that's our motivation, and with that, I'll go ahead and get started with the first session. So the funding for our research in the health effects of drug-drug interactions comes from the National Institute on Aging and the National Institute of Diabetes and Digestive and Kidney Diseases. So in terms of methods to study the health effects of medications, I should mention that Charlie and I are both faculty members at the University of Pennsylvania in the Center for Clinical Epidemiology and Biostatistics and in the Center for Pharmacoepidemiology Research and Training, which is where our team is based. In terms of potential conflicts of interest, here I've listed all the pharmaceutical consulting I've done in the past 2 years. As far as I know, none of this is directly relevant to the work that I'm going to be presenting today. So the outline of my presentation today, which I hope will end at 45 or 50 minutes past the hour to allow time for questions, is first I'm going to provide some background and introduction for the need for pharmacoepidemiology. Then we're going to spend some time going over pharmacoepidemiology research methods, and then finally give an overview of some of the data resources that can be used for pharmacoepidemiology research. And again, if you have questions as we go along, feel free to put them into the chat box, and I should be able to see those. So the first point I'd like to make is that in the United States in particular, drug use is ubiquitous, so much so that if you sample water out of the Great Lakes, you're going to find metformin. If you take a group of older adults, defined as those 65 years of age and older, and you ask them how many unique prescription drugs they took in the last month, only 10% of them will say 0, and close to 40% of those will say 5 or more. And these data are from 2005 to 2008, and I'm sure that drug use has gone up since then. So with this high degree of polypharmacy, there are lots of opportunities for potential drug-drug interactions, and drug-drug interactions aren't always identified prior to drug approval for a number of reasons, including that, particularly for old drugs, we don't know everything that there is to know either about their pharmacokinetics or their pharmacodynamics, so they're difficult to predict. And prediction exercises sometimes over-predict. It's difficult to know what the clinical and health relevance is from predicted drug-drug interactions. So I'm a pharmacist, and I have epidemiology training, and I put those things together and conduct pharmacoepidemiology research. And pharmacoepidemiology is something that I had never heard about in pharmacy school. It took till I was out in practice till I learned about it. And it's defined as the health effects of drugs and other medical products, including biologicals and medical devices in populations. And the thing that gets me really excited about pharmacoepidemiology are really twofold. So one is it's studying pharmacology, and I'm interested in the biological effects of drugs in humans. And so this basically uses humans as probes to study pharmacology, which is, you know, I went to pharmacy school originally because I was interested in pharmacology and thought I wanted to be a laboratory science researcher. And the second aspect of pharmacoepidemiology that interests me is that almost by definition, you're studying clinically important health outcomes, that is, measurable effects on people that have real impact, like getting hospitalized for serious hypoglycemia, or getting hospitalized for serious bleeding, or having a stroke or other thromboembolic event, rather than studying effects on biomarkers or other variables that may or may not have clinical importance. Pharmacoepidemiology is also the science underlying the public health practice of drug safety surveillance. And there are large numbers of people both in the pharmaceutical industry and in regulatory agencies like the Food and Drug Administration, whose job it is to try to conduct surveillance on potential adverse effects of drugs after they're approved into the market, and pharmacoepidemiology is the basic science underlying that public health practice. There are a couple of metrics that we use in epidemiology in general and pharmacoepidemiology in particular that it's worth talking about and distinguishing. Two of those that are related but distinct are risk and rate. Another word for risk is cumulative incidence, and the definition of risk is the probability that an event will occur over a specified period of time. So an example of this would be the results of a cohort study where you found that 1.2% of subjects experienced hypoglycemia. Of course, that 1.2% cumulative incidence or risk is only interpretable if you know what the follow-up period is. And the follow-up period is likely to differ among patients. So if you know the average follow-up period is 7 months, then that helps you interpret the 1.2% risk or the 1.2% cumulative incidence. A rate is another word for an incidence rate, and the definition of that is the number of new events in the study group divided by the amount of person time. And an example of this would be in a cohort study, and we'll talk about what a cohort study is in a minute. The rate of serious hypoglycemia can be found to be 2 per 100 person years, or another way of saying that is 2% per year. So we notice here that the denominator is in terms of person time rather than people. And when you hear this, it's also helpful to know how long people were followed for, because oftentimes the incidence rate changes as a function of the duration of follow-up. So for allergic reactions, for example, they tend to happen early in therapy, and people who survive the first 2 or 3 months of therapy are less likely to get an allergic reaction than people at the beginning of therapy. And actually, that declining a trend for a declining rate of adverse events in a group of people as they're followed over time is true both for idiosyncratic events like allergic reactions, and it also tends to be true for dose-related events, as long as the dose-relatedness isn't related to cumulative dose. So obviously there are some adverse events like cardiotoxicity associated with anthracycline cancer chemotherapy agents where the rate of events is associated with cumulative dose. That's sort of the exception to the rule. In general, the rate of adverse events in a treated population tends to go down over time. So the main point of this slide is to distinguish risk from rate because different epidemiologic study designs yield effect measures that use different metrics. So some will present a relative risk or a cumulative incidence ratio, whereas others will present a rate ratio or a hazard ratio that has person time in the denominator. So that leads us to, so those are measures of occurrence. Measures of occurrence are in a particular group. Usually in epidemiology studies, we compare exposed people to unexposed people, or people who are getting drug A to people who are getting drug B. And in order to make comparisons, we have some measure of association. Sometimes these measures of association are called effect measures. When you call them effect measures, that means that you're making some causal inference that the exposure caused the adverse event, or the beneficial event for that matter. And the two most commonly used ratio measures of association are the risk ratio and the rate ratio. And not surprisingly, the risk ratio is the ratio of two risks or two cumulative incidences, and the rate ratio is the rate ratio of two incidence rates. So that's also called the incidence rate ratio. So the risk ratio is also called the relative risk or the cumulative incidence ratio, and that's defined as the risk in the the risk in the exposed group divided by the risk in the unexposed group. Or if you're comparing drug A versus drug B, it would be the risk in people getting drug A divided by the risk in people getting drug B. So for example, the risk ratio for hypoglycemia in a cohort study that compared glyburide to glipizide might find that the ratio of risks is 1.4 comparing glyburide to glipizide, meaning that it's 40% higher in the glyburide group than the glipizide group. And in epidemiologic studies, we convey the statistical uncertainty around whether that around the precision around the point estimate of 1.4 by presenting a 95% confidence interval. So for example, the 95% confidence interval could be between 1.2 and 1.5. A rate ratio is also known as an incidence rate ratio. It's similar to a hazard ratio. And the rate ratio is defined as the rate in the exposed divided by the rate in the unexposed, or the rate in people getting drug A versus the comparator drug. So for example, the rate ratio for serious hypoglycemia comparing glyburide to glipizide can also be 1.4. And most of the time, the risk ratio is going to be very close to the rate ratio, although there are unusual times when they can be different in meaningful ways. A couple words about causation. So in any epidemiologic study, including randomized clinical trials, we make inferences about causality, but we never actually observe causality. So a common saying is that association is not causation, and that's true, but in all empiric science, unlike math, we always infer causal effects and we never observe causal effects. Making causal inferences or causal judgment, causal inferences always involves judgment, so there's no absolutely defined causation, and that's true even with randomized trials. So randomization generally increases the strength of the causal inferences, but randomization is neither necessary to make a causal inference, nor is it sufficient to make a causal inference. So for example, practically no one now refutes the conclusion that smoking causes lung cancer and that smoking causes heart disease, even though the most compelling evidence of this is epidemiologic. There aren't any randomized controlled trials where we took people and randomized them to smoke cigarettes or not smoke cigarettes, and then look at the rates of adverse outcomes. And then the flip side of the coin is that a randomized trial doesn't necessarily guarantee that you're observing causation. If that were true, then all randomized trials would get the correct answer guaranteed, and you would need to conduct only one randomized trial to answer any question, and meta-analyses of randomized trials would be superfluous, and we know that they're not. So that's it for the introduction. I'm now going to move on to an overview of pharmacoepidemiology research methods. So pharmacoepidemiology research methods can be divided into two major groups, descriptive studies and analytic studies. So we're going to start with descriptive studies on the left, case reports, case series, and ecologic studies. So a case report is just that. It's an anecdote. It's an anecdotal report of the symptoms, signs, diagnosis, treatment, and follow-up of an individual patient. A case series is a collection of related anecdotes. And as Daniel Patrick Moynihan, former senator from the state of New York, would remind us, the plural of anecdote is not data. So just because you have lots of anecdotes doesn't mean that you have a strong basis for causal inference. Most of the time with case reports, the implied control group is what would have happened to that person had they not gotten the drug, but we don't know what would have happened to that person had they not gotten the drug. We're just making the inference that the bad outcome that we observed in them would not have happened if they didn't get the drug that we're blaming the adverse event on, but we don't actually know that. That said, case reports and case series are very important in drug safety surveillance, and we rely on clinicians in particular to make astute observations about what they see in their practice and to make these hunches that this drug may have caused this adverse event. And the hunches are right often enough that they're useful to identify hypotheses or signals of potential drug safety problems. And sometimes regulatory agencies need to act before controlled evidence can be obtained. So sometimes regulatory actions are made on the basis solely or mainly on case reports and case series. So an example of a published case series is from the Food and Drug Administration related to cisapride and fatal arrhythmia. And the utility of this case report was that it identified the potential association between cisapride and ventricular arrhythmias, some of which were fatal. And case reports and case series are very helpful in helping us to design, to formulate hypotheses and helping us to design pharmacoepidemiology studies. So for example, they can help us clarify the outcomes to study. In the case of cisapride, we would want to look at torsade de pointe if we're able to look at that. We could look at, well, we know that 7% of cases died. We know that 28% were resuscitated. We know that arrhythmic cases from cisapride were often manifest as syncope or fainting. Case reports and case series can help us identify potential risk factors for the event that we may want to include as factors in our study, either as potential confounding factors, if these factors are likely to be more commonly present in people who got cisapride than whatever control group we want to look at, or maybe they're effect modifiers, they're subgroups in which cisapride may be more likely to have an effect than in other groups. Case reports and case series have limits to their utility, particularly for making causal inferences. And the first one is one that I mentioned earlier. We don't know what would have happened in the absence of the suspect drug. Other limitations include that the anecdotes are incomplete and potentially biased in the ascertainment of them. So we certainly don't know about all bad things that happen to people while getting drugs. And we hear that the FDA adverse event reporting system is one of the disadvantages of it is underreporting. I think that the problem with using the term underreporting is that it implies that we can fix it by increasing reporting. And I don't think we're ever going to get to complete reporting, nor do I think that the FDA spontaneous reporting system could possibly handle complete reporting. I think that we're probably drowning in reports now. I think that we may be better off having fewer, better documented reports than many poorly documented reports. So, and the next disadvantage is that the reports are frequently poorly documented. The diagnoses can be wrong. You can't use anecdotes to calculate incidence because you don't have all of the events and you may not have the size of the exposed population. And spontaneous reporting databases have limitations in the ability to identify an increased risk of an otherwise expected event. So for example, rofecoxib, a COX-2 specific nonsteroidal anti-inflammatory drug, was used in patients with rheumatoid arthritis. Patients with rheumatoid arthritis develop heart attacks, and it would be difficult to identify an increased risk of an otherwise expected event like heart attack in a population like people with rheumatoid arthritis using spontaneous reporting systems, and all of these are reasons to conduct pharmacoepidemiology studies. Nevertheless, case reports and case series are very valuable for identifying hypotheses and helping us to design controlled studies to evaluate those hypotheses. In addition to that, so this is a pie chart of sources of information that contributed to drug safety labeling changes by FDA in the year 2010. And despite all the not so nice things I said about spontaneous reports, they were responsible for more than half of all drug safety labeling changes. And pharmacoepidemiology studies, which is what I do day in and day out, was responsible for only 6% of drug safety labeling changes. So this was back in 2010 before FDA's Sentinel program was as active as it is now. And my hope is that with the so Sentinel is a large capability that gives FDA the ability to do pharmacoepidemiology research. My hope is that with that capability, the slice of the pie that's due to spontaneous reports will shrink, and the slice of the pie that's due to pharmacoepidemiology studies will grow in terms of contributing to knowledge of drug safety. So now we're going to move over to the right-hand side of the diagram and talk about analytic studies. and I'm going to start with randomized clinical trials. And I'll tell you in advance that my approach to teaching analytic pharmacoepidemiology designs is to emphasize the similarity among the designs rather than to emphasize the differences among them. If you understand a randomized trial, you can understand a cohort study because they differ mainly in the fact that randomized trials are randomized and cohort studies are non-randomized. And if you understand a cohort study, you have an easier time understanding a case control study because you know that case control studies are really just cohort studies where you're not taking everybody who did not develop the outcome, you're sampling from people who didn't develop the outcome. Self-controlled studies are a little bit different, but I try to fit them into the same framework. So let's move forward and talk about randomized clinical trials. So in randomized clinical trials, you have a study population, you have some random process like the flip of a coin that determines who gets treated and who gets untreated, or who gets treatment A versus who gets treatment B. And we follow people forward in time, and some people develop the event, that is, they have this lightning bolt. Some people don't develop the event. An example of the randomized trial design used in the context of post-approval drug safety is the VIGOR trial, which was a comparison of upper gastrointestinal toxicity of rofecoxib versus naproxen in patients with rheumatoid arthritis. And the background for this study was that, as people on the call probably know, non-steroidal anti-inflammatory drugs, at least at the time that COX-2 inhibitors were developed, were responsible for an epidemic of gastrointestinal bleeding and ulceration, and were the cause of about 100,000 hospitalizations and 16,500 deaths per year, and many of those occurred in older adults. So COX-2 specific inhibitors were developed specifically to provide analgesia and anti-inflammatory beneficial effects to people who need them without the gastrointestinal effects. In fact, it had been demonstrated prior to approval that on the basis of endoscopy, you stick a tube down someone's nose or throat and you look at their stomach, that there are fewer endoscopically observable ulcers in people getting rofecoxib compared with non-steroidal anti-inflammatory drugs. But what wasn't known was whether that led to a difference in the rate of clinically observable episodes of serious bleeding and peptic ulcer. So the research question was, does rofecoxib compared to naproxen result in fewer clinically meaningful gastrointestinal events? And so they conducted a randomized clinical trial of 8,000 patients with rheumatoid arthritis who were age 50 and above, or you could get in if you were 40 and above and were on chronic steroids, randomized to either rofecoxib or naproxen. And they found that the rate of gastrointestinal events was indeed lower in the rofecoxib group compared to the naproxen group, 2.1 per 100 person years in the rofecoxib group versus 4.5 per 100 person years in the naproxen group. You divide one incidence rate by the other incidence rate, and you get a rate ratio. In this case, is about 0.5, with the confidence limit that excludes the null value. So the null value for ratio measures of effect is 1, and if the 95% confidence limit excludes the null value, then we say that chance is an unlikely explanation for this result. This trial also looked at the rate of myocardial infarction and found that the rate of they reported the cumulative incidence of myocardial infarction. So here in the same slide, we have both incidence rate and cumulative incidence, and they found a cumulative incidence of 0.4% in the rofecoxib group and 0.1% in the naproxen group, and that, if you do the math, comes out to a rate ratio of 5 with a 95% confidence limit that excludes the null value. The investigators, the authors of the paper in the New England Journal, chose to invert this rather than showing a positive association with rofecoxib. They reported it as an inverse association with naproxen, and argued that their results were consistent with the theory that naproxen had coronary protective effect, and highlight the fact that rofecoxib does not provide this type of protection. Subsequent studies have supported the conclusion that rofecoxib actually increases the risk of heart attack. So moving from randomized trials to cohort studies is a relatively small step. So in a non-randomized cohort study, we have a study population, and then we have a non-random process that determines who gets treated and who gets untreated, or who gets treatment A versus who gets treatment B. And this non-random process encompasses a lot. So it's everything from who goes to the doctor versus who doesn't, what kind of doctor people go to, and doctors certainly prescribe differentially to different kinds of patients. So the big disadvantage of cohort studies compared with randomized trials is that before, with randomization, at least if you randomize enough units, on average we had assurances that the baseline characteristics of the treated and untreated people were similar, whereas with non-randomized cohort studies, we don't have that assurance, and there can be baseline differences in both measured and unmeasured factors. And a lot of what we do in non-randomized pharmacoepidemiology is try to account for and control for potential baseline differences between treated and untreated patients, or people getting treatment A versus treatment B. On the other hand, a relative comparative advantage of non-randomized cohort studies or randomized trials is that randomized trials, as we'll go over, often aren't done in a group of patients that's very representative of the group of people that will ultimately get the drug. So randomized trials can produce correct results, but they may not be very generalizable. So an example of a non-randomized cohort study that was used to study drug safety in the context of, in the post-approval setting, was a study published by Bill Cooper and colleagues looking at serious cardiovascular events in children and young adults using drugs for attention deficit hyperactivity disorder, or ADHD. The background for their paper was that at the time ADHD drugs were used in more than 2.7 million children in the US. My guess is that that number has gone up. These are stimulants. Some of them are amphetamine-based, and we know that those drugs increase heart rate and blood pressure. And we know that increases in heart rate and blood pressure have been associated with increases in cardiovascular events. There were also anecdotal reports of seemingly healthy kids who experienced unusual for kids adverse events, including cardiac death, heart attack, and stroke. And those kids were receiving ADHD drugs. However, on the other hand, there are also cases of children who experience sudden cardiac death, heart attack, and stroke, and they're not taking ADHD drugs. So you can't make the inference that the ADHD drugs cause the unusual events in kids. Due largely to case reports and case series, mixed amphetamine salts were banned in Canada in 2005, but were later reintroduced. So the research question that was addressed in this study was, do ADHD drugs increase the risk of cardiovascular events in children and young adults? So what they did was they used pre-existing healthcare data from 4 health plans. In doing so, they identified about 400,000 users of ADHD medications between the ages of 2 and 24, and about double that number of unexposed people who were matched based on health plan, calendar, year, age, and sex. And the endpoint that they studied was a composite endpoint of sudden cardiac death, heart attack, and stroke, and they had central adjudication of outcomes. And here's what they found. So this is a bar graph where the vertical axis is the adjusted rate of that composite outcome expressed in terms of per 100,000 person years. And 100,000 person years is a big number, and the reason that the results are expressed for 100,000 person years is that it's in children and young adults, and the frequency of cardiovascular events is low in children and young adults. So what they found was, so they looked at 3 groups. So they looked at current users of drugs for ADHD, they looked at former users of drugs for ADHD, and they looked at non-users. The reason that they looked at former users was to address the potential concern that maybe something about ADHD itself changes the underlying rate or is associated with the underlying rate of cardiovascular events. So by comparing users to non-users, we're comparing people with ADHD to people without ADHD. So former users are probably people who at least at some point had ADHD. So the thought was, and I agree with the rationale, that the former users may look more like the current users than the non-users. So what they found was that if anything, the rate may have been a little bit lower in the current users, but the rate was comparable across all 3 groups. These results have been replicated by other groups, and as a result, at least on a population basis, there's not the same concern for cardiovascular events being caused by ADHD drugs as there used to be. So let's move over to case control studies. So case control studies are basically cohort studies where we sample from among people who didn't develop the outcome. So like a cohort study, there's a study population. Like a cohort study, there's a non-random process that determines who gets treated and who doesn't get treated. And then instead of studying everybody, so we study everybody who developed the event, regardless of whether they're exposed or not, and we take a random sample of the people who didn't develop the event, again, without regard to whether they're exposed or not. If you choose your potential controls at the time that each case develops the outcome, then the measure of association that you get from the case control study, which is the odds ratio, is interpretable as the rate ratio. And this is the usual way that people do case control studies. An example of a case control study used in pharmacoepidemiology is one of my favorite studies looking at drug-drug interactions. This came from David Yerling's group in Ontario. And the background for this, and my guess is that many people on the call are familiar with this paper. So we know that trimethoprim sulfamethoxazole inhibits cytochrome P450-2C9, which metabolizes glyburide. We know that clarithromycin inhibits the function of P-glycoprotein, which pumps digoxin out of the gastrointestinal tract and the central nervous system. And we know that potassium-sparing diuretics and angiotensin-converting enzyme inhibitors both inhibit the secretion of potassium in the urine. And the research question is, or was, do these potential drug-drug interactions have clinically observable consequences? So what David Yerling et al. Did was they used data from the Ontario Health Plan, which provides universal coverage to people 65 and above in Ontario. So they took them a year later once they turned 66. So what they looked at was, among person time exposed to glyburide, is the rate of serious hypoglycemia, so that they, within person time exposed to glyburide, they identified cases who experienced hypoglycemia and controls who didn't experience hypoglycemia, and looked at the frequency of prior trimethoprim sulfa in the cases and in the controls. Similarly, in people getting digoxin, they identified people who were hospitalized for digoxin toxicity. Those were the cases. The controls were people who did not develop digoxin toxicity while on digoxin, and they looked at the frequency of clarithromycin use in the cases and controls. Similarly, they took person time exposed to potassium-sparing diuretics. They identified instances of severe hyperkalemia leading to hospitalization during that person time. Those were the cases. The controls were randomly selected people who, while they were taking potassium-sparing diuretics, did not experience an episode of severe hyperkalemia. And then the study looked at the frequency of ACE inhibitors in the cases and compared that to the frequency of ACE inhibitors in the controls. here were the results that they found. So the vertical axis has the adjusted odds ratio, which in this case is interpretable as the rate ratio because of the way they sampled in their case control study. So they found that if you're getting trimethoprim sulfa, methoxazole, you're 6.6 times as likely to experience severe hypoglycemia if you're also taking, if you're taking glyburide, you're 6.6 more times as likely to experience severe hypoglycemia if you're getting trimethoprim sulfa compared to if you're not. If you're taking digoxin, you're 11.7 times as likely to develop digoxin toxicity if you're taking clarithromycin than if you're not. And if you're taking a potassium-sparing diuretic, you're 20.3 times as likely to experience severe hyperkalemia if you're getting an ACE inhibitor compared to if you're not. And finally, I'm going to go to self-controlled studies. So self-controlled studies are newer study designs. These were developed in the 1990s, and they have some advantages and some disadvantages compared to other controlled designs, which is mostly cohort studies and case control studies. So in self-controlled designs, the only people in the analysis are people who have had the event. These are also called case-only designs. So each case serves as her or his own control. So it includes only people who have experienced the event that you're studying. And what the framework is it compares the event rate in people, among people with the outcome during exposed times compared with during unexposed times. And by doing this, it inherently controls for factors that remain constant within the person over time, that is static factors, although it doesn't inherently control for factors that change within the patient over time, or dynamic factors. On the other hand, if those dynamic factors that you want to adjust for are measured, you can adjust for them in the analysis. The self-controlled designs ask the question, why is this event happening to this person now rather than at a different time? Whereas cohort and case control studies ask the question, why is this event happening to this person rather than other people? Self-controlled designs or case-only designs are good for studying transient effects of acute and intermittent exposures where you know the exposure time and you know the risk window, that is the time over which the exposure could plausibly have an effect. There are two major self-controlled designs, the self-controlled case series and the case crossover design. I'm going to give an example of the self-controlled case series design. So this was a study that examined on time versus delayed early childhood vaccination with the outcome of interest being seizures. The rationale for this study was that an increasing number of families are delaying childhood immunizations because of fear of adverse events or because they don't want their child to get 5 needles on the same visit to the pediatrician. The Institute of Medicine and the National Academy of Sciences has called for an assessment of the safety of non-standard immunization schedules. So the research question of these investigators was, does delaying vaccine administration affect the risk of vaccine-induced seizures? We know that vaccines are safe, but they're not completely safe, and they are associated with some risk of adverse events, including seizures. So the setting that this was conducted in was using healthcare data from managed care organizations, and the people in the study, or the study subjects, were children who had at least one physician visit, plus an emergency department visit, or hospital admission for a seizure. And the positive predictive value of the algorithm for identifying seizure is in the 90%. So that is, if your algorithm says that somebody had a seizure, there's a 92 to 99% chance that if you get their hospital chart will confirm that they had a seizure. This is a pretty high positive predictive value when it comes to outcomes that you would study using health data. So these are the results right from the table. And just to orient you to the table, they looked at 3 different vaccines, MMR, measles, mumps, and rubella, varicella, and MMRV, measles, mumps, rubella, and varicella. And then for each vaccine, they looked at 2 different time periods of administration. Either it was on time, it was late, it was on time, and it was late, it was on time, it was late. So over here, the IRR is the incidence rate ratio, the thing that I was calling the rate ratio. So we see that for MMR vaccine, if you give it on time, the rate ratio for seizures is 2.65. If you give MMR vaccine late, the incidence rate ratio for seizures is 6.5. So by delaying vaccination, you increase the rate ratio from 2.6 to 6.5 for MMR. For varicella, if you give it on time, it's 2.75. If you give it late, it's 3.64. For MMRV, if you give it on time, it's about 5. If you give it late, it's about 10. All right, so that concludes the section of the talk on research designs and pharmacoepidemiology. I'm just going to spend a minute or two on data resources that could be used for pharmacoepidemiology. So there are spontaneous reporting systems, that is collections of anecdotes. You can collect data in an ad hoc way, so you can conduct your own cohort study by enrolling patients who start on a drug and follow them forward and see what happens to them, and you can identify adverse events either by looking at their insurance claims or by calling them up on the phone or by having them check into an online surveillance system. You can use pre-existing healthcare data. Pre-existing healthcare data come in two general characteristics, either electronic medical records or administrative billing data. I have examples of both listed. There are similarities and differences between them. So the main differences are, with electronic medical record data, they could be more granular on the one hand in some senses because you can have access to physician notes. On the other hand, they could be incomplete because somebody might go to multiple physicians, and if they don't share the same electronic medical record, then you may not know about all of the drugs that they're prescribed and all of the things that happen to them. The other major difference between electronic medical records and administrative or billing data is that electronic medical records tend to have drugs that are prescribed by the physician rather than those that are dispensed by the pharmacy, whereas administrative billing data tend to have drugs that are dispensed by the pharmacy. So in an electronic healthcare record, you'll know that somebody received a doctor's prescription for something, and you may then know the number of refills that the prescriber allowed for, but you won't know whether they picked up even a single dispensed prescription of it, let alone the third or fourth. Whereas in administrative billing data, you know when the data were dispensed from the pharmacy, which is one step closer to actual ingestion compared with a physician prescription. So at this point, that ends the presentation. So I'm going to stop here and watch the chat box for questions that people might have. And either people don't have any questions or I can't see any questions being submitted. Okay, so somebody asks, would it be possible to get a copy of your slides? So yeah, I think I can make a copy of the slides available on the CPERT webpage. CPERT stands for Center for Pharmacoepidemiology Research and Training. If you just type CPERT into your browser, you should come to our page, and then there's a section for resources. So we will make copies of our slides available there. And if we figure out how to save the recording of this webinar, we'll also save a copy of the webinar there. Second question. So you recommend we use electronic systems with administrative data. It really depends on the research question. So one of the main limitations of using pre-existing data to study drug-drug interactions is that you need to make sure that the health outcome that you're interested in studying can be captured using administrative claims data or whatever data that you have. And there are some health outcomes of drug-drug interactions that are well captured, such as bleeding, such as thromboembolism, such as serious hypoglycemia, but there are other outcomes that probably aren't so well captured, like serotonin syndrome. I have no idea how I would study serotonin syndrome using electronic health data. So it really depends on a lot of factors, including your research question. Another question is, how about SureScripts data? I don't know what SureScripts data are. They could be, so I know that there are other commercially available sources of data, and SureScripts may be one of those. To cover all bases, I'm not sure what that question means. All right, any other questions? So there are 2 other sessions in this series. I forgot to put the dates. You know, this third slide isn't updated. Let me go back to the first slide. We're going to be sending out a request for comments. So we'd like suggestions on if we were to do webinars in the future, what would people like to hear about? For example, we could present webinars on additional findings of pharmacoepidemiology studies of drug-drug interactions, if people think that those would be of interest. We'd be happy to consider presenting other topics as well. So if you have other ideas, actually, so regardless of whether you have ideas or not, please respond to the questionnaire about what you thought about the webinar, and we'd love to hear about your suggestions. Pharmacoepi Journal Club could be fun. Yeah, we could do that. All right, so I think we're going to go ahead and end the webinar. Thank you very much, everybody, for joining us. This was a big success. Have a great rest of your day.